Yeah.
Instead of showing the video, I'm just going to introduce you to SAAMBR, the South African Association for Marine Biological Research. We're a non-government organization, and this year we're celebrating our 70th anniversary, which is quite something for an NGO in South Africa. We operate 3 divisions. The first is the Oceanographic Research Institute, and Bruce and Ryan are part of the Oceanographic Research Institute, or ORI. That's where our scientists undertake applied marine research, primarily along the East Coast. We are the people that look after Oshaka SeaWorld, which is probably Durban's main tourist attraction. And then we run the Oshaka SeaWorld Education Center, which inspires people to care for the ocean of all ages. So that's just a very brief introduction to SAAMBR. And now it's my pleasure to introduce our first speaker for tonight, Dr. Ryan Daly. Ryan has been with SAAMBR not for very long, but he has a very long history in the field of marine research. He's a very accomplished photographer, accomplished man on the boat, and also a very, very sharp scientist who produces some incredible papers and has an amazing whiz ability with data. So Ryan, I'm going to let you share your screen, and over to you.
Thanks so much, Judy. It says host disabled participant screen sharing. Can you just let me share my screen?
Yep, I've enabled it.
There we go. Got him. Okay, brilliant. All right. Thanks so much, Judy. Can you all hear me? Or can you?
Yes, we can hear you. Yes.
Great. Thanks. Good evening, everybody. Sorry, I look a bit under the spotlight here because of load shedding. I feel a bit under the spotlight, but let's talk about some giant trevally, or GTs as they're more commonly known. Certainly research on these incredible fish has been a passion of mine for many years now, and I've been able to share this research with many equally passionate people, all in various countries in the West Indian Ocean. And just, yeah, I'll share with you guys a little story about these incredible fish. So I guess the first question is, why giant trevally, or GTs as we'll probably refer to them going forward? For those of you who do fish, if you've ever hooked one of these fish, you'll have no doubt why they are just such charismatic fish. But even for those who don't fish, the species has certainly inspired many people to film them, find out about them, and over the last decade, they've really put South Africa and the West Indian Ocean on the map. This top photograph is of them shoaling, that was featured in Planet Earth many years ago from the BBC. And this bottom photo is of the more recent Blue Planet 2, where the GTs were filmed eating terns out of the air in the Seychelles, which, if you can imagine a fish seeing a flying bird through the water-air interface, it really is an incredible feat, and certainly highlights their incredible abilities as top predators, and also emphasizes their unique role that they play as top predatory fish in the marine ecosystem. So really, I mean, if that still leaves you guessing why these fish, they also reach over 80 kilograms. They're these massive top predatory fish, and they are known to be higher on the food chain than sharks. And I see Jessica Glass is with us here too, and some of her research proved that they do indeed have a higher trophic level than some sharks, which is just incredible for a large predatory fish. They can also be long-lived, reaching 25 years, and typically they have a tropical Indo-Pacific distribution from Hawaii throughout the tropical Indo-Pacific. And in South Africa, where we are down at the southern edge of their distribution, they do occur in South African waters, more so in Isimangaliso, where we have more consistently tropical areas, but they do also occur all the way down to the Breda River, in fact, where someone caught one this year, which is the new record for the distribution in South Africa. So the question, I guess, is how important is South Africa for GTs? And more than that, do they migrate, considering that South Africa is at the edge of their distribution? Are they moving into and out of South Africa throughout the year. If they migrate, where do they go then? And what could drive them to migrate? Is it only the temperature in South Africa, environmental factors pushing them to migrate, or is it something more? And ultimately, I guess the main question here with our research on these incredible, charismatic, but also ecologically important fish is what does it mean for conservation? So I'm just going to tell you a quick story, I guess, about our research on them over the last few years and what this means for their conservation. So our first question, do they migrate? I guess, how do we answer this? So we've relied on tagging them with acoustic tags. This is an example of an acoustic tag. It's just a small tag that we implant into their body cavity, and that pings a unique code that's picked up by receivers, as you can see on the top screen here. This underwater receiver then logs the tagged fish's code, and we download that about every year to get data on it. And in order to get data on their migrations, we really rely on a community of people overseen by the ATAP network out of SAAMBR to share data on these tagged fish, to share data from their receivers all along the coast, from the west coast of South Africa all the way up to Basruta, Vermisi Island, northern Mozambique. So we're able to answer the primary question of the study, which is, do they migrate? So this is just a quick study site map to put us all on the map. Our main site that we tagged these fish was in southern Mozambique, where it says aggregation site there, where we had a number of receivers looking at their fine scale habitat use. But then we also put receivers along the whole coast, and we also tagged them in South Africa, primarily in Ismangli. So just so we didn't tag them all at one place, but we wanted to see what the majority of these fish were doing in South Africa. So to start, what we found was that at our aggregation sites, which we filmed and dived with for close to 10 years now, is that this is the largest aggregation sites of these fish in the world. So every year, approximately 5,000 of these mature fish aggregate at the study site. And it's difficult to get your head wrapped around it. That's just a photo of what some of the fish look like, but it really is a significant amount of fish. We calculated that it was over 30 tons of GT that aggregates every year at the study site. So what does this mean? Well, they're not there the whole year round. Certainly that many GTs would probably eat every single fish on the surrounding reefs. So they need to go and find food at other times of the year when they're not there. So our first hint sort of to answer the question about do they migrate is the fact that they are in one place seasonally, but where are they when they're not at this aggregation site? And if we look at the data of their occurrence, we've got just the time on the x-axis and the number of tagged GTs on the y. That red line represents temperature, and you can see that that's just the seasonality of the water temperature throughout those 3 years represented. And this is just evidence to show that they're definitely there in early summer, but they are not there in late summer or winter at all. So again, more reason to answer the question, well, where do they go when they're not at this aggregation site? So slowly started to put pieces of this puzzle together at the start of the study years ago. And what we found ultimately was that these fish are indeed undertaking these massive migrations. And bear in mind, before our study, one of the furthest migrations of GTs with acoustic telemetry around the world was only 28 kilometers. So what we found was that these fish are doing up to 650 kilometers one way down to Port St. John's, but then they come back up to the aggregation sites every year. So at some point, they're swimming up to 90 kilometers per day along the South African coast. And this 90 kilometers has been sustained all the way from southern Mozambique to the south coast of KZN. So that's 90 k's per day for at least 5 to 7 days straight. This also means that these fish are undertaking this migration for up to 5 years now. So we've been tracking these fish for more than 5 years, and they have these repeat patterns of migration along the South African coast. Each fish does have a unique migration pattern. So this is just a subsample of the whole, but just really incredibly showing that these relatively small fish are able to migrate these massive distances every year. So if they're traveling so far along our coast, there must be a good reason for it. And what we found, looking more closely at the data and the research, was that they specifically cue in to aggregating at the full moon. So this is just some of our data showing that the highest abundance of these fish at this aggregation site is exactly over the full moon, and fish are more likely to arrive just before the full moon and depart just after the full moon. And when they're there, they seem to undertake these color changes, all associated with spawning. And for those of you who know your fish biology, if you're a fish in the open ocean and you need to spawn with other fish, you need to arrange a time and a place to do so. And this is called a Gynecomastic broadcast spawning strategy. So these fish have decided that at the start of every summer, the first full moon, or indeed the first 3 full moons at the start of every summer, is when we all going to get together at this one particular place and at this one particular time. So these fish have cued in incredibly well to seasonal cue and a lunar cue to arrive at the same place at the same time every single year. And between this, they're undertaking migrations to other areas where they most likely feed and meet up with other GTs along the coast. So just starting to figure out what these fish are doing on our coast. What this means is that the majority of South African giant trevally are going to southern Mozambique every single year to spawn, which is very significant because it means that these fish could be vulnerable to fishing pressure or other threats across our border, which means that no matter what we do in terms of conservation for them in South Africa, they could still be very vulnerable just across our border. And we really need to think about this in terms of working together with our neighboring country to ensure that we have congruent management policies for these fish and ultimately to ensure their conservation. And I guess what next? These fish continue to be fascinating, and we've got a lot of new research in the pipeline. This includes tagging them in Seychelles. We've already had some interesting recent research published from Seychelles looking at their home range and how it may take these fish up to 36 months to use the entire extent of their home range. We've also started tagging them in Mozambique, Lazruto and Vamizi, but we hope to look at the differences in populations along the Southern African coast to look at if fish elsewhere are doing anything different. We've also got some exciting projects such as putting Fitbits on them. This top photograph is a GT with an accelerometer tag looking at the activity patterns specifically related to spawning. So we really hope we can find out more about that. And ultimately, we are working very closely with Southern Mozambique, where this aggregation of giant trevally occurs, and luckily it's in a marine protected area just across the border, and they understand the importance of this aggregation of giant trevally, and we've got a really good working relationship with management across our border, and we hope that we can improve or at least keep our current conservation management plans working for these fish so that we can continue to enjoy them on our coast in South Africa. So yeah, in a nutshell, that's what we've been up to. And I think we'll have some questions after Bruce's talk, if that's right, Judy. But thanks so much, guys, for your attention tonight.
Thank you so much, Ryan. That was great, and yeah, absolutely amazing what these GTs are doing. 90 kilometers a day and sustaining that is simply incredible. So what we're going to do now is we are going to move on to Bruce's presentation. So Bruce will be talking to us about the evidence of the effectiveness of MPAs. And then after his presentation, we'll go into the questions. So Bruce, because you're not here, I will share your presentation. And if you can just tell me when you want me to change the slides. And if you go too slowly, I'll just change the slides for you anyway.
Okay. Okay. Good evening, everybody. And yeah, thank you for making the time to come and listen to us chat this evening. And yeah, certainly thanks, Ryan, for a excellent presentation. It's certainly been one of the most exciting projects that I've been involved in with tracking these GTs. But yeah, tonight I want to talk to you a little bit about what I've been doing for the past 20 years, which is looking at trying to look at the effectiveness of some of our marine protected areas, both in KZN and the Eastern Cape. Thanks, Jude. Right, so they're two long-term projects. The first one is in the Ismangaliso MPA in northern KwaZulu-Natal, and the second one is down in the Ponderland MPA in the Eastern Cape. And my hope is that tonight, at the end of the talk, I would have convinced you all that marine protected areas really do work. Thank you. All right, I think most of you are aware that we've had an increase in our marine protected area network in 2019. We've had 20 new MPAs added to our network, but we're still at only 5% of our exclusive economic zone is found in formally protected marine areas. Tonight, the two that I'm going to talk to you about are Ismangaliso up here in the north to start with, and then the Ponderland MPA down here, which I think are still our two biggest coastal MPAs in terms of size. I stole this from a paper that I've been working on with George and Steve Kirkman and other co-authors, but basically just setting the scene here that MPAs, as I'm going to call them, marine protected areas, are widely advocated as a means of conserving marine species and ecosystems and allowing recoveries of depleted populations and communities. I think that's pretty well known now. But an important point is that the ecological efficacy needs to be critically evaluated. The way we do this is normally based on the status of populations and communities inside versus outside MPAs before and after proclamation. So what I'm going to share with you tonight is examples of doing just this and trying to look at this efficacy. Right, and I've been up here today looking at exactly that, a very similar picture I took on my camera today. This is the Ismangaliso Retton Park. Looking down the coast, that's Leven Point in the distance. So the first project I'm going to be telling you about is the results of experimental research fishing and tagging in this area over the past 10, 20 years. Thank you. Okay, a little bit of history. The St. Lucia MPA was established in 1979. That was then incorporated with the Maputoland MPA into what we now call the Ismangaliso MPA. But this one has been here for a long time, and it's basically extended from Cape Vidal here in the south up to White Sands, just north of Sodwana in the north, and the middle area between Red Cliffs and Leven Point, a distance of about 25 kilometers, was a sanctuary area. So what we wanted to do was compare the inshore fish communities between the no-take sanctuary area, this area, with those of the exploited area south of Leven Point. But I just started the project in 2001, and the beach speckle ban came along, which meant that there was no more fishing allowed north of Cape Vidal further than people could normally walk. So I had to quickly change the objectives of the project to compare this area south of Leven Point, which was, I call, the previously exploited area, against that of the sanctuary, using the sanctuary as a benchmark. So that was really what happened in the study. And the way we did it is we had 4 areas, 2 areas south of Leven Point in the previously exploited area, EA and EB, and 2 areas in the sanctuary, SA and SB. Each one of these areas was 2 kilometers of coastline, marked off at 100-meter points with numbered flags. And we fish this area with a group of 4 anglers and 1 vehicle on the first day, and then fish the SA area on the first day with a group of 4 anglers, and then alternate over 4 days, so that we'd all fished all 4 areas over a 4-day period using the same bait, the same tackle. And that was how we made the comparison between the sanctuary and the previously exploited area. Thank you. So the aims of the project really were to compare catches of shore angling fish within the St. Lucia Marine Reserve Sanctuary and to monitor if there was recovery over time. To determine movement patterns of these species on a fine spatial scale using tagging, because we're now looking at movements of 100 meters rather than sort of kilometers, to attain estimates of growth rates of fishes from the tag recapture, and to also look at the optimum size that these inshore no-take areas needed to be to provide enough protection. Thank you. Okay, so just basically how we did it, we used two 4x4 vehicles with 4 anglers in each. You might recognize Tom Hecht and Denham Parker there. We used standardized fishing tackle, my great friend Pat Garrett and Simon Chater there fishing with me. We used circle hooks mainly, and those were made barbless, so we crimped the barb flat, and we used standard baits, mainly things like squid, sardine, mackerel, prawn, that sort of thing. Next one. I put a lot of emphasis onto good handling of fish. So as soon as the fish was landed, it was placed into a bucket of fresh seawater. It was then measured on a stretcher with a measuring rule down the middle. And you'll see I put a wet cloth over the fish's head, covering the eye, which helps to calm the fish down and protect it from damage. If the fish was big enough, over 30 centimeters fork length, then we tagged it using an Ori dart tag into this area here, and then immediately release. So we tried to keep the fish out of the water for no longer than 30 seconds. Thank you. So the fish I'm going to talk to you about tonight, just giving you a little bit of a species ID here. From top left, grey grunter, speckled snapper, and large spot pompano were the 3 most common fish that we caught in this project. But lots of others. Other important ones were Natal stumpnose, blacktail, cave bass or lanternfish, big eye stumpnose, cat-faced rockcod, and yellow belly rockcod. Thanks, Judy. Just to show you some results now, and I'm just going to simplify it and show you results from the first 10 years. So this is November 2001 to July 2011. In that time, we caught over 12,000 fish during 50 field trips. We were doing four trips per year. When we started, we were doing 6 trips per year. We tagged over 5,000 of those fish and had a recapture rate of 13%. So 700 of those were recaptured. Those included 87 different species from 38 families. And now we at 116 species from 42 families. So it's increased even more in the next 10 years. But just to show you that we're comparing apples with apples, if you look at the composition here, you'll see grey grunter, speckled snapper, and large spot pompano are the 3 most common fish caught in both areas, except the difference being that speckled snapper were much more abundant in the sanctuary area than in the exploited area there. And also just look at these numbers here. In the sanctuary area, over 7,000 fish caught, whereas in the previously exploited area, just over 5,000. Thanks, Judy. And I think this graph really tells the story of what happened. If you look at the green bars, you'll see the catch rate. This is catch per unit effort, which we use as an index of fish abundance. And you can see that over the 10-year period, it changed very little. Obviously, it fluctuates from trip to trip. Often the summer months, we have higher catches than the winter months. But if you look at that mean there, it stayed around 0.5 fish per angler per hour. If you look what happened in the previously exploited area, south of Leven Point, when we started, the catch rate was only half of what it was in the sanctuary area. But over time, we've seen this gradual increase in abundance of fish in the previously exploited area. And if I'd had time, I could have showed you how this graph has actually just kept on going up. And in the next 10 years, those 2 lines have actually met each other. So now the catches south of Leven Point are as good as they are in the sanctuary area. Thank you. So yeah, there's just too many species to show you results from every one. So I just cherry picked a few examples. this is what happened with grey grunter. Again, we've got the years on the x-axis, and we've got catch per unit effort on the y-axis. And you can see in the previously exploited area in this first graph, so this is the area south of Lemon Point, we got the significant increase in the catches of grey grunter, and then they sort of leveled off. Whereas in the sanctuary area, there was no significant change. They just sort of fluctuated around this mean. If you look at this graph, you'll see in the two previously exploited areas, EA and EB, you'll see that there was a big increase between when the project started in 2002 and 2011, and the same in EB, whereas in the sanctuary, they stayed very similar. This just shows seasonality of grey grunter, which shows that they're not seasonal. You get them right throughout the year in the same sort of abundance. Thank you. So now looking at the mean size of another fish, the speckled snapper. Again, if you look at what happened in the previously exploited area, we went from just over 250 millimeters being the mean size, and it just kept on increasing and got bigger and bigger until 10 years later, we were looking at a mean size of 350. So these fish just increased steadily throughout that period. In the sanctuary, interestingly, we see this little dip here. They went from an average of 350 and dipped down to 300, but up to 350 again. We think this was really a recruitment event when we had good spawning of speckles and lots of small fish moving into the sanctuary area. That's what caused that. But again, if you look at EA and EB, the two previously exploited areas, when we started and towards the 10 years later, big increases, whereas in the sanctuary, very similar. And a little bit of seasonality here, showing that these fish feed more aggressively during the summer months and a little bit slower during the winter months. Thank you. Then if we look at the movement of these fish. So what I've got here is the distance moved on the x-axis and the proportion of recaptures on the y-axis. And I'm just going to show you a few of the species, starting with grey grunter, 57 recaptures. They show very little movement. Most of the fish are recaptured in the first 200 meters. Most of them actually don't move at all, but some of them will move up to just over a kilometer. If we go to catface rockcod, same sort of picture. Most of them highly resident, but some of them are actually moving quite far. Same sort of story with yellow bellies, very, very resident, but a small population of them, about between 5 and 10% are moving. Same story with the cave bass, very resident, but some moving. And then with the speckled snapper, which is my sort of real hobby horse and my favorite fish, same sort of thing, but look at the number of recaptures, 652. So this is an enormous data set to base this kind of study on. So it shows you that these fish are highly, highly resident in the inshore reefs and the surf zone along this coastline, but there is a small part of the population that does at times abandon their home range and move into other areas. Okay. So what we could use this information for, and this slide is the subject of a whole paper on the subject, but I'm just trying to simplify it here. So we quantified the home range for each of these species that we were working on by taking the 95th percentile of the intra-study site movements. So we just looked at those graphs that I've just showed you and worked out the 95% percentile and excluded the movements where they abandoned their home range and didn't come back into their home ranges. So that was how we quantified it. And then to estimate how big a no-take area should be to protect these populations, we simply doubled the home range size in all directions. So if you can see this here, if that was the home range, that middle circle there, it's doubled in all directions. That would give you an area suitable to protect a population of this species. And on a linear sort of coastline, like the coast that we were working on, you just simply multiply by 3. So you take the home range and multiply by 3. So looking at all the fish that we were working on, I came up with an estimate of a minimum no-take area of between 3 and 6 kilometers of suitable habitat. So that's reef habitat along the coastline. and then replicated every 15 to 20 kilometers. So that's to ensure that there's connectivity between the populations, either through movements of adults between the areas or via egg and larval drift, the fish spawning and then drifting and settling into adjacent areas. Thank you. We also were able to look at the growth of speckled snapper because we just got so many recaptures. I could use this information to work out the growth. You measure them when you tag them, and you measure them again when you recapture them. And from that, you can work out the growth rate. So this growth curve here basically shows that when the fish are small, at around 30 centimeters, when we started tagging them, they were growing at about 40 millimeters per year. But the time that the fish reach big old speckles here, we call them double-decker or triple-decker speckles. When they get this size, they're growing at about 20 millimeters a year. So they grow really, really slowly. And if you just think that a 600 millimeter fish has been growing at that rate for a number of years, you can realize that these fish can reach ages of 30 years or more. Thank you. So yeah, using this information, we were able to do some quite interesting comparisons. We could take the growth rate of speckled snapper that had been tagged and recaptured just once. If you think about it, a fish being caught and tagged is quite stressful. And I thought that this might be affecting their growth rates, so that the fish that we caught on multiple occasions, so caught and tagged and then recaptured more than once, some of them up to 7 or 8 times, individual fish, that would affect their growth rate. But when we did the comparison, there was no significant difference between the growth rate of single recaptures and multiple recaptures, which is good for the way that we're handling these fish. We're catching them, releasing them quickly, and not really impinging them too much. Similarly, when you're using bait, often the hook is swallowed. And we recorded whenever a hook was swallowed and cut off. We didn't try and dig that hook out. We just left it in place and clipped it off. But because it was barbless, they normally come out quite quickly. We compared the growth between fish that had been tagged and recaptured and swallowed the hook with those where the hook was normally removed. And again, we found no significant difference in the growth rate, which is again a positive result for the way that we are handling these fish. Thanks, Judy. So using the information that I've shared with you, we were able to approach the management authorities, KZN Wildlife and the Isimangaliso Wetland Park Authority, and make some suggestions on how to rezone the MPA to make it more effective. And what we did was we kept the no-take sanctuary area between Red Sands and Leven Point, which is a 25-kilometer stretch. And what we did is we added on a buffer zone. And here we allow catch and release fishing, but no take. So there's no harvesting. You can't take fish home here, but fishermen can walk to this area or cycle to this area, enjoy magnificent fishing, but release their catch. And then at the control zone, so at Sodwana Bay itself, where there's a access point, here you're allowed to catch your fish and keep them according to bag limits and size limits, and exactly the same at Cape Bidal. So by doing this, the no-take sanctuary area has a strong buffer zone on either area, which will make this area more effective. Thank you. Thanks, Judy. So some quick conclusions. I think we've seen and I've shown you that there's been a strong recovery in fish abundance and size in the area south of the sanctuary since the beach vehicle ban was implemented in 2002. Most of our surf zone fish have relatively small home range sizes, less than 2 kilometers, but some fish do move and abandon their home range. This can give you an example of seeding or spillover of adult fish. Some surf zone fish, such as speckled snapper, grow very slowly and can live for over 30 years, which makes them extremely vulnerable to overfishing. And then the no-take areas, we estimated between 3 and 6 kilometers of coastline length, consisting of suitable reef habitat, are required every 15 to 20 kilometers to ensure adequate protection and connectivity between populations. So some of the results of this project have been incorporated into the recommendations and have been implemented in the new zonation for the Isimangaliso Wetland Park in 2019. Thanks, Judy. So I was supposed to have 20 minutes for this talk, and I know I'm going to go over, but I'm going as fast as I can. I'm now going to whip you down to the magnificent Ponderland Marine Protected Area in the Eastern Cape. And this is a picture of a waterfall bluff, which is one of the unique areas in the world where waterfalls drop directly into the sea over the escarpment. Thank you. A little bit of history here again. Nkambati Marine Reserve was proclaimed in 1991 under the Transkei Environmental Decree. The first ORI proposal for establishment of a large MPA in the Ponderland region was made in 1998. This area was seen as a big gap in the sort of biodiversity protection along the coast. Between 2002 and 2003, ORI was involved in conducting a big marine biodiversity survey of the coastline. And fortunately, in 2004, Vali Musa was ready to leave the Minister of Environmental Affairs position, and he wanted to go with the flags flying. And so we were able to get the proclamation of Ponderland done really quickly. In 2005, we spent time developing a management plan for the MPA. And then between 2006 and now, we implemented a monitoring program to try and evaluate the effectiveness of the MPA. And that's what I'm going to share with you a little bit tonight. Thanks, Judy. Basically, to monitor the recovery of protected reef fish populations in the Ponderland MPA, to compare the catch per unit effort, which I've told you is linked to the abundance of fish, the catch composition, the different types of fish, and the size structure, the size of the fish caught on selected reefs within the no-take zone, with that on similar reefs in the exploited zone. So again, it's a comparison between areas. To determine fine-scale movement patterns using tag recapture and to look for evidence of spillover, and then to compare the overall fish community within the no-take zone with that of similar reefs in the exploited zone using underwater visual census. So here we got in and actually counted fish underwater. So a quick map to show you where we are. We're in the Eastern Cape, just south of the border with KwaZulu-Natal. The Ponderland MPA extends from the south bank of the Mzamba River down to the north bank of the Mzimvubu River at Port St. John's. And for boat-based exploitation, it's very simply zoned into an exploited zone where fishing is allowed in the north here, the yellow, and in the south, and then a big no-take zone. That's about 40 kilometers between the Sekombe River and the Mboiki River. And it extends 10 nautical miles or 10 kilometers out to sea, approximately to the 1,000-meter depth contour. So what we did for the comparison here was to find 4 similar areas of shallow reef habitat, one at Mzamba here, one down here off Nyameni, one off Mtentu there, and one off Nkambati itself, off Gwekwe Point. And we did a comparison between the exploited area and the no-take restricted area by doing research fishing and diving in these areas to compare the fish populations. Thank you. So I was fortunate to have some colleagues from the Council of Geoscience who were able to do a side scan sonar for me of my research blocks. So that's one of my blocks off Gwekwe Point, exactly 2 kilometers long and 1 kilometer wide. So it's a 2 kilometer square block. And they did some side scan sonar in here to look at the reef habitat. All this dark gray shaded area is where the reef habitat is. And I went through this very carefully, and every little crack or bump or hole where you might find some reef fish hiding in unnumbered. And the way we fish this area is that I just generate random numbers on these reef points before each trip, and then we come and fish those points using a GPS. We just and we'll use an anchor just above the point, and then fish the point for at least 20 minutes and no more than an hour, and then move to at least 3 different points in that area each trip. Thanks, Judy. Again, we use a controlled fishing survey. So I use 3 anglers and Scarborough type reels, 80-pound braid line, bottom sticks. Our traces I make them all up beforehand. I use an 8-0 circle hook on the bottom and a 6-0 circle hook on the top. And these are crimped for barbs, and bait is basically chokka or squid and sardine are the main baits. Thank you. Similar to there's a Mangalisa project. I try to invite a guest angler on every trip, and this exposes people who've never fished in a protected area and fished in an area where the fish populations are close to being pristine, because I can show people as many graphs and I can talk to them as much as possible, but seeing is really believing. So that was an important part of this project, was inviting these guest anglers. Thank you. Thanks, Judy. Right, the nice part, catching the fish. We did catch some beautiful fish in this area. If the fish were big, I landed them in a stretcher like this to protect them. If they were small enough, we would just literally swing them aboard. They went straight onto a wet mattress. There's a foam mattress underneath here. Again, the wet trough over the eye, mouth closed to measure it, carefully measure it, and then tag it with the dart tag. Thank you. Next one. I think there's a delay. Next slide, please, Judy. So if the fish, because we were fishing between 10 and 30 meters depth, if they were blown, as happens when you bring the fish up from barotrauma, we'd use a hypodermic needle, which is inserted into the swim bladder just to release that pressure so that they could swim down. In cases with big fish like black mussel cracker, we actually used a sea equalizer, which is a downrigger type mechanism, which is attached to a big weight, and you just let that weight take the fish down. The pressure of the water recompresses the swim bladder, that pops open, and the fish can swim away unharmed. If there was no barotrauma, we simply released them from the surface, and then obviously all the information was recorded: the size, the species, the tag number, position, etc. Next slide, please. Okay, then we did the diving surveys. We only did these annually because of the difficulty of getting good weather and good visibility in this area. It's renowned for, it's not called the Wild Coast for nothing. So lots of big seas and lots of dirty water. So normally our underwater visual census was done in May and June, which is the sort of calmest, best time of year for this work. Next slide, please. We started off by doing point counts, because that's how we did the original biodiversity survey. But then we moved across to doing strip counts, where we would lay out a 50-meter line on the reef and compare fish on either side and record their sizes. Next slide, please. But because of the difficulty of diving in these areas and getting suitable conditions, we moved over to using baited remote underwater videos, which is just simply a stainless steel frame like this with a little GoPro underwater camera mounted on it, a bait arm, and then 1 kilogram of mashed sardine in the bait arm to attract fish in. And this would be left on the bottom to record for an hour, so we could see what fish were on the reef. Next slide, please. That's just a picture of the bruv on the reef. There's the bait canister, and here's a whole shoal of slinger that have been drawn towards it. And right here is a black mussel cracker or punskop, which one of the fish that we've actually tagged. There's his tag sticking out there. Next slide, please. So very quickly, results. 40 field trips were completed between 2006 and 2016. So that's a 10-year period that I'm going to tell you about. Over 8,000 fish caught from 88 species, 27 families. We tagged over 4,000 of those, of which 700 capture rate. If I include multiple recaptures, it goes up to 27%, which is unbelievable. I don't know of any other study in the world using dart tags that's got a recapture rate like that. And that's already telling you something about our results. In terms of underwater, we did 96 point counts between 2007, 2010, 24 strip transects between 11 and 12. And then we moved over to the bruvs in 2013 and did over 100 bruv deployments. Looking at the fish composition, these are the most important fish: the Natal sea catfish or barbel, very poisonous spines, half moon rock cod because a little half moon on its pectoral fin, beautiful Scotsman, an important reef predator, the black mussel cracker, also very well known, popular angling fish. What used to be called the blue emperor, but now has changed its name to Natal emperor because it's actually an undescribed species, which a paper is busy being written on it. Yellow belly rock cod slinger, the most important commercial fish in Natal. Flap-nosed hound sharks and catface rock cod. So those are some of the most important fish we caught. Thanks, Judy. Looking at the catch composition, again with the controlled fishing survey, first thing you'll notice, again, much more fish caught in the no-take area than an exploited area. And in the no-take area, the catches were dominated by important lionfish species. So things like slinger, Scotsman, yellow belly rock cod, black mussel cracker, etc., dominated our catches. Whereas in the exploited area, much smaller fish and dominated by things like blue emperor, which we call scavengers because they're just so many of them, and lots of small slinger. Interestingly, more catface rock cod and half moon rock cod in the exploited area. Next slide, please. So I think, again, this graph tells you the story from the very beginning. Every year that we've fished, these are 4 trips per year, all amalgamated into one. We've caught almost double the number of fish inside the no-take sanctuary compared to the exploited zone. That's fish per angler per hour on that. And that was where the park was proclaimed in 2004. So, very clear evidence that the biomass or abundance of fish is much higher in the no-take area. Next slide, please. So when you look at it on an individual species, it's even more apparent. So that's slinger. Again, this is catch per unit effort or fish per angler per hour on the y-axis and years going along on the x-axis. 5 to 6 with Scotsman. Same story with black mussel cracker. Same story with yellow belly rock cod. Interestingly, it switches around for catface. We caught more catface outside, and again, more half moon outside and more blue emperor or Natal emperor outside the no-take area. Again, sea catfish about the same in both. Next slide, please. So if you look at this in terms of size now, I think for just about every species, it's so clear. This is fork length on the x-axis here and percentage frequency on the y-axis. Again, you can see much bigger fish in the no-take area. So the slinger were huge. I mean, we were catching 35, 40 centimeter fork length slinger in 10 meters of water, which is unheard of along the rest of the coastline. Same story with the Scotsman, much bigger fish. Same story with black mussel cracker. Again, yellow belly rock cod, very clear. Catface not as clear, but although we caught less of them, generally they were bigger. Again, same sort of story with half moon, slightly bigger fish. Blue emperors or Natal emperors bigger, and even the barbel tended to be bigger in the no-take area. Thanks, Judy. Next slide, please. So quickly, some results of the tagging now. So we tagged over 4,000 fish with, if you look at multiple recaptures, over 1,000 recaptures. Next slide, please. So looking at movement, this is the distance moved in meters on the x-axis. And you can see just again, almost the same kind of pattern as we saw up in the Simangalusa. Highly resident. Fish moving, up to 400-500 meters, but not much further than that, and a small percentage of them moving out of the area. So very, very resident reef fish species. So quite high numbers that we're looking at here too. 77 slinger, over 200 Scotsman, a little bit more mobile, but still having this resident component and a small component moving. Same story with just about every case. Yellow belly is extremely resident, same with the catfish. Half moons, we didn't get any that moved sort of long distances, very few Natal Emperor recaptures. And again, with the sea catfish, very resident and no fish moving out of their home range, just staying there. Next slide, please. So just to show you an example of one yellow belly rock cod that we caught. This rock cod we named Rocky, and he was caught just south of Gwekwe Point over here on this little reef in about 16 meters of water. And we caught the same fish 9 times, so 10 recapture points. And every single one of those capture points were within that 577 square meter area. So that shows you, and that's over a period of 4 years, it shows you how resident these fish are, just staying in the same area. That's his home. Next slide, please. And in looking at the Scotsman, as I said, were a little bit more mobile. This is my sampling area off Mtentu. You can see that the fish are moving a little bit more within the area. That's about a distance of probably just under a kilometer, about 800 meters. There were some reef pinnacles here and here that they moved between. But a lot of the fish we recaptured, over 80% of them in the same area. But about 10% of them were actually leaving the MPA and being recaught up the KZN coast. We in fact had most recaptures from the Richards Bay area, from commercial fishermen who reported the tags here, and we've had 3 that have moved all the way into southern Mozambique. So there is a component of the Scotsmen that are leaving the MPA and moving north, and this could be an example of spillover with these fish seeding the adjacent areas. We also believe these fish are moving north to spawn, with spawning mostly taking place up in northern KZN and Mozambique. All the fish down here, the Scotsmen, are actually not breeding. So this might be part of a northward spawning migration. Next slide, please, Judy. Underwater visual census, a little bit more difficult to see, but similar numbers counted in both areas. Again, you can see the dominance of slinger in the no-take area and the dominance of fish like Scotsman, so important lionfish, whereas outside we've got smaller fish, smaller Natal emperors, slinger, blacktail, that sort of thing. So lots more smaller fish outside. Thanks, Judy. Next slide. If you look at the trophic structure of these fish communities, it becomes more clear. So if you compare Piscivores, so those are fish that eat other fish. So again, these would be Scotsman and yellow belly rockcod, much more abundant in the no-take area compared to outside. Similar with the other species, but with Planktivorous, you see the reverse, much higher in the exploited areas. And these are little fish. These are little fish like damselfish, little goldies, that type of thing that feed on plankton. So these are typical prey fish species, which are much more abundant in the exploited area. So you've got a real change in the fish community. Next slide, please. Next slide, Judy. Thank you. Right, quickly conclusions. So I think you would have seen from this presentation, there's good evidence of the value of the no-take area in protecting a greater abundance. There are a lot more and much bigger commercially important lionfish species. The tagging data has provided us useful data on growth and movement patterns, and we've used that to quantify their home range size. The immigration of tagged fish from the no-take area has given us an idea of some of the spillover into adjacent exploited areas. And the underwater visual census and the BRUVs have shown us that the fish community in the no-take zone is dominated by these larger piscivorous species, while the exploited area is dominated by smaller prey fish species. So that's also quite an important finding from this study. So I think overall, just to share some outputs of both projects, for me, one of the important things is that 2002 volunteer anglers, I think 130 something on the Ismangaliso project and about 70 on the Ponderland project have assisted with doing the research fishing over the past 20 years. And many of them have become ambassadors for MPAs because they've actually seen with their own eyes the difference in the fish populations, the abundance and the size. We've had a number of graduates coming out. I managed to get my PhD on this Ismangaliso project, and Jade Maggs did his master's on Ponderland. Had 16 scientific papers, 2 book chapters, 11 technical reports, 29 talks and conferences, maybe that's 30 now, 21 popular articles, and 5 television documentaries done on these two projects. We had a revised management plan for the Ponderland MPA in 2012, which used some of the results of the monitoring. And similarly, as I showed you, we had a complete rezonation of the inshore zone of Ismangaliso Park in 2019, which was also based on some of the results of this project. Next slide, Judy. So yeah, that's my talk, and I'd just like to really thank all of those anglers that have assisted on these projects over the time and contributed their time over the last 20 years to these various organizations, SAAMBR, DEF, Ismangaliso Wetland Park, and Eastern Cape Parks for funding, Sharks Board and Ezemvelo KZN Wildlife for logistics support, and Peter Breton and Clive and Nikki Filler for accommodation in Port Edward, and the Marlin Ski Boat Club for helping us with launching. Thank you very much, and thanks everyone for listening. Thanks, Judy. Oh, one more slide. There's one more slide, Judy. Viva MPAs, viva!
Thank you very much, Bruce, for that synopsis of an incredible amount of research. I'm sure that there are some questions. I know some people have put questions into the chat. And I know, Ryan, thank you very much. You've answered a lot of those questions. But I think, let me just see if there are any in the chat that we need to go through. There's one question that's, how much spillover have you recorded for species like speckled snapper beyond Mapellan, Bruce? That's just a question from, I think it must be from Rudy.
Okay, so very little. It's quite incredible what a biogeographic break Cape Vidal is. If you look at the fish composition south of Cape Vidal, the dominant reef fish predator is catface rockcod, whereas if you move north of Cape Vidal, it becomes speckled snapper. So they really are sort of more tropical species. And as you go further south from Cape Vidal, their numbers diminish very, very quickly. So we've actually caught very few as far south as Mapellan. But most of their movement and the spillover of adults takes place in a northerly direction, with adults moving northwards rather than southwards.
I think now I'm just going to let anybody ask questions. So if you want to put up your hand and ask questions, yeah, just Jeremy.
Thanks, Judy. A couple of questions. First of all, to Bruce, to what extent is poaching by ski boaters from launch sites like Port Edward a significant problem in the Ponderland MPA? And then 2 issues with for Ryan. What percentage of the GTs actually head north towards the equator when they leave Ponto rather than heading south into South African waters? And then the other thing I'm battling to get my head around, and that is the difference between nomadism and migration. In other words, are the GTs when they leave Ponta actually migrating southwards, or they're just nomadic and that they're drifting southwards, and when they all decide to return to Ponta, then that is what one would constitute as a migration? Thanks.
Okay, I'll go for the first question. Jeremy, it is a problem, and we've been trying our best with very little resources to keep control of this. At this stage, the actual poaching seems to be on a fairly low level. It's a number of commercial boats mainly that operate out of Port Edward that sneak in there on nice days when the sea is calm. We know who they are. We have apprehended some of them, but it is a challenge, and certainly we are kind of using peer pressure and anglers on the water who are reporting them to try and apprehend them, because at the moment we don't have any boats or law enforcement active in the area. So it's really through other anglers that are assisting in the process and telling us about it, and then we mobilize some sort of enforcement response. But fortunately, I think at this stage the poaching is still at a fairly low level, but we need to obviously keep watching it at all times.
I think Tiki and then Peter.
Thanks, Judy. Just a quick question, Bruce. You flew over St. Lucia. Was the mouth closed? Could you see?
I'm gonna keep you in suspense, Nikki and Tiki, and then let Ryan quickly answer those two questions.
Oh, okay.
Thanks, Bruce. Yeah, some good suspense. Jeremy, thanks for your questions. Some tough ones for Tuesday evening, but I'm going to try and provide some insights. Yeah, absolutely. When we tagged the GTs in southern Mozambique of sort of Ponta, we expected half to go south, half to go north. And initially we detected hardly no GTs going north. And we originally thought that was due to a lack of receivers in the north. But over the last few years, we've had more receivers just north of the study sites, and still we get hardly any GTs going north. So GT tagged at the aggregation and within South Africa are pretty much all going south, which is really interesting because we would expect more to go north. And those that did move north, one or two individuals really only moved less than 10 kilometers north. Relating to the question about migration versus nomadism, another really good question. And I think what we see in the data is that DGTs are queued into very particular locations. So for example, some other guys were asking about Mtentu. Some fish tags at Mtentu estuary where they aggregate will swim specifically from Mtentu on the wild coast in the Eastern Cape to southern Mozambique and back again multiple times within a few months. In one summer season, they'll go to southern Mozambique for the full moon and down to Mtentu for the half moon and then back again. So these are very directed migrations. As I said, 90 kilometers per day, every day, very directed, very specific, very cued into particular times and places for those fish at least that we have tagged and monitor. I hope that helps answer that.
Okay, I'll come in and break the suspense. Yes, the mouth is closed, Tiki. It closed about a week ago, but we flew over the system with Greg and Richard today, and it's absolutely brimming. It's prop full. The system is looking beautiful. The mouth it's not very high above the sort of the high tide mark, so I think we'll still get overtopping that type of thing with big seas. So there hopefully will still be some recruitment into the system taking place, which, as you know, becomes more important as we get closer to spring, when a lot of the fish are spawning.
Oh, that's great to hear, Bruce. And fantastic amount of work. That was one, I don't know, horrifying project in a way. I mean, the amount of work that went in there was superb, but I think you've got some really great stuff out of it. Well done.
Thank you.
Thanks, Sticky. Peter's got a question, and then I'm going to try and do some of the questions from the chat. This is more difficult than it looks.
Hey, thank you very much for the impressive presentations. I think for me, it's Peter here from IUCN. So I see here, this is like really impressive showcase of how scientific output can inform management design, which is really exciting. And so some few questions to Bruce, just 3. I wanted to understand why some fish abandon their home ranges, at least to know whether it's random or there's certain underlying, maybe physiological or environmental factors involved. Then the second question is for areas where you showed for certain MPA areas where people can catch and release. I think you've referred to the baits that you use in your study that are carefully selected. So I wonder whether there are mechanisms to control or maybe to oversee the types of baits like individuals use in their catch and release areas to minimize harm to the fish. Then the last question is, so you pointed out, I think maybe Ryan pointed out that MPAs in South Africa are around 5% of the EEZ area. So in order to fast track the establishment maybe of additional MPAs in South Africa, do we need to conduct these or maybe similar multi-year studies, or can we just use like results from your study to maybe design new MPAs based on maybe abundances of certain species in certain localities along the South African coast? And then to Ryan, I just wanted to know, like the GT, do people eat it, or like what is the ecological significance of this particular fish? Thank you.
Okay, thanks, Peter. I'll try and remember those questions. The first one was about why do these fish leave their home range? So a home range is an area that a fish lives in and where it forages and does its thing. And it's quite difficult to understand why they leave. In some cases, we have quite a lot of sand movement in the inshore zone, and we have reefs being covered with sand. And that would obviously force a reef fish to actually move and find somewhere else. But those generally tend to be quite short movements and not very far. It's been a lot of thought given to this, and it seems to be that a lot of reef fish are basically born either genetically as stayers or as goers, and about 5 to 10% of the population seems to be born with a gene that forces them or makes them want to travel. And these fish are able then to move much greater distances. And if you think about it in an evolutionary point of view, it's quite a good idea, because you won't get your population isolated in one little reef and then sort of dying out. You'll get that movement. So it seems to be a strategy that's been found in many reef fish species to have a certain percentage of the population being more nomadic than the others. The next question was on
the bait.
On the bait. Okay. So what we've done for our catch and release areas is we haven't specified what bait can be used, but what we have done is we've specified a number of controls. So all fishermen have to use barbless hooks, for example, so crimping the barb, because that has been shown to greatly increase the survival rate of fish that are caught and released. And also we would stress the good handling of fish that are going to be released to ensure that they have better survival rates. But yes, it is possible. In some areas, you can actually restrict the use of bait completely, and you can say no baits, but only lures, for example. You can restrict the type of hooks. You can say only circle hooks or only single hooks, not treble hooks. There are lots of different options that can be used in that case. The third question was on
whether we need to undertake multi-year studies like you did to do an additional MPA, or is this a template?
So basically, yeah, these can certainly help and feed into it, but when you're looking at biodiversity protection over the whole EEZ, you obviously want to maximize the number of ecoregions and the number of habitats and species that you include in your protected areas. So we've adopted in South Africa a kind of process of planning and trying to find out exactly where our different ecosystems are and our different marine communities, and trying to ensure that we get a small proportion of each one of those protected in our network of MPAs. So it's no good just having one big MPA up in Isimangaliso and then nothing down in the Southern Cape, where you've got a completely different ecosystem with cold water and kelp compared to warm water and corals, for example. So we're trying to ensure that our protected areas have representatives of all those different ecoregions in them. So the work that we're doing can certainly feed in, and there's a bigger picture that one needs to look at when designing and implementing a network of protected areas.
Thanks, Bruce. I think that there was also a question from Peter for Ryan. Have you got that, Ryan?
Yeah, thanks, Peter. I think asking about the importance of the GTs if they're targeted to eat, and certainly they're an important fishery species, specifically in Mozambique. They're an aggressive predator, so can be caught by net, hook and line, baited fishing, but they also are exceptionally important as a high value catch and release species in the region, and globally. They support many high-end fly fishing and other sports fishery catch and release and targeted fishing, because they're probably one of the most prized game fish. So they are important commercially to eat, commercially for fishing charters, and also ecologically.
Thanks, Ryan. I'm just going to go to a couple of the questions in the chat. From Njabula, the scientific evidence of MPAs is very interesting. Has there been any study on the socio-economic benefits of these MPAs concurrently with your research? And then the second question is, does the abundance of planktivorous fish inside MPAs indicate high abundance of plankton in these MPAs?
Okay, Judy, I'm going to pass the first question on to you, because having recently written a paper on the subject, I think you're well equipped to answer it.
Okay, thank you for that curved ball, Bruce. Okay, so when it comes to the socio-economic impact of MPAs, the research is lagging behind some of the biological evidence for the effectiveness of MPAs. It's a project that various people have tried to look at it over time, and most recently there is quite a large project being run through WWF to really understand, and the government, to understand the socio-economic impact and value of MPAs. And it's work that has started, and it's going to be continuing. So we do have a number of studies on the go at the moment. We're writing a few papers on the subject. And I think it's something that's going to become increasingly important, understanding the role of MPAs and people. So we can understand MPAs in nature, but it's really looking at MPAs and people and the impact of MPAs on people, and how can we make sure that people benefit from those MPAs, because that's the only way we're really going to be able to get the support that we need for the MPAs to succeed. And just as a matter of interest, today we are launching South Africa's first ever Marine Protected Area Day. On the 1st of August, we will be holding South Africa's first MPA Day. So watch out, join us. And the reason we're doing that is because we did some research in the aquarium, and I think that probably all of you know from personal experience, when you talk to people about a marine protected area, they say, a marine protected what? Over 80% of our visitors could not name one marine protected area in South Africa. So we really need to highlight the role of marine protected areas in conservation, what they do, what they can do, and then start to really provide the evidence of the socio-economic effect of MPAs on people. So thank you for that question, and that I hope has answered it. Bruce, do you want to talk about the planktivorous fish?
Okay, I think, yeah, basically it for me, what the results were was that we had a lot more piscivores in the no-take area than outside. And so we had a situation, what is often called prey release, because those big predators, the big Scotsman, the big yellow belly rockcod, are the first fish that are caught by line fishing in those exploited areas. So they're removed from the population. When you remove those predators, you have that sort of automatic increase in their prey species. So that was what we were witnessing, rather than it being any change in the amount of plankton in the water or anything like that. So I think that was sort of really important for me. And this instance of no-take areas where you've got good, strong protection and having much more predators is very, very, very visible in many well-protected areas around the world. And I think of some of the coral atolls and islands in the Pacific that haven't ever been fished. They are completely dominated by large predators. So you have, unlike where you have in a terrestrial protected area, like a game reserve, you'd have a sort of pyramidal type shape where you'd have very few top predators and lots of herbivores, lots of impala and wildebeest and zebra, and very few lions. In marine systems, it appears that very well protected systems, pristine systems, have a high number of predators, and it's because they live so long. Those predators are often long-lived, and the sort of production in the ocean is so strong that it can support that sort of high biomass of predators. But yeah, it's a very interesting scenario and different from our terrestrial systems.
Okay, thank you. I've got a question here from Div. We get many reports of poaching in the Mtentu. Should we not try and revive the catch and release fly fishing venture with community benefits? A question for both Ryan and Bruce.
Thanks for that question, Div. Yes, there are a number of people inquiring and looking at that right at this very moment. Our recommendations have been based on our experience of catch and release and of the Mtentu fishery itself. And this is directly from Ben Pretorius, who ran the Ufudu fly fishing catch and release fishery in Mtentu for 7 years in the sort of late 1990s, early 2000s. Is that what would be much better is a kingfish watching tourism operation. This is an absolutely unique migration of fish into Mtentu, where we don't know of anywhere else in the world that it happens, where you get up to 1,000 adult kingfish or GTs going into the system. They seem to be sitting in the surface layers of Mtentu. So they're in the fresh water above the saltwater wedge in the estuary. And we believe it's probably for parasite control. They're probably going in there to get rid of external copepod parasites in the gills and so on. They don't seem to be feeding there. They're just doing these huge daisy chains meandering around in the surface. And it's absolutely unbelievable to sit and watch this type of behavior. So we feel from a conservation point of view, what would be a much better and stronger and more sustainable practice would be to try and get the local communities involved in kingfish watching, whereby you promote it, you market it, and you take people to witness. You sit in the cliffs above the river looking down at these fish, and you have an experienced guide explaining what's going on. We do have 3 acoustic receivers in Mtentu at the moment. And as Ryan explained, we actually monitoring their movements in and out of the system, trying to understand how long they remain in the system for. and that kind of thing. But yeah, that would be, for me, a much better suggestion. And both Ben and I have been sort of suggesting this to the guys who are trying to promote catch and release to rather go for kingfish watching. So I don't know if you want to add to that, Ryan.
No, thanks, Bruce. I think it makes sense.
I've got another question here from Div. Thanks, Bruce and Ryan. Dwesa and Kwebe, chiefs and headmen, have requested presentations on the benefits of MPAs. Would you like to do your presentation there on the 31st? I think I'll let you take that question out, but I think that, Div, the important point is that we really have to communicate the research, the results of the research, and what's happening more widely. So I think that your point is well taken, and we've got to get out there with the results of the research much more effectively. Question here. Your research has proven that MPAs are vital to revive depleting populations of fish species. Do you think it is possible to get more protection, as the idea was 10% by 2020, and unfortunately this didn't happen?
Yes, I think that is what South Africa is going to strive for. We certainly do want to increase the percentage of protection, and there are a number of projects that are starting to look at that. However, I feel personally, and many agree with me, is that we've got 20 new, or what we must do first is we've got to manage those properly. We've got to get management plans in place, and we've got to get them working. Once we've achieved that, then we look at bringing on the next 5%. So it's not to say that the two can't work concurrently, but our first priority is to manage the MPA network that we've got well and effectively, and then to look at the targets of increasing the amount of habitats that we're protecting.
Okay, thanks, Bruce. I've got one last question from Sharif Patrick, and then we'll probably have to close. Night fishing for most of the time has never been policed. During the winter months, Geelbeek and Dogger salmon are caught far more than what commercial fishermen catch. So there's a question about that. Is there anything that Bruce can put forward to slow this down or to stop night fishing at all?
Okay, it's a good question. We've been very concerned about the exploitation on Geelbeek and dusky cob in particular over the years. Fishermen have learnt where their spawning aggregations are, and they go to meet them every year. I've been out there with fishermen as well and seen what happens. We've also got lots of shark predation or depredation on those fish now. The sharks have learned basically about this. So you're getting huge numbers of fish being eaten off the line. So they're healthy, good, strong fish that anglers are hooking, but not getting them out the water because the sharks are taking them. As a consequence, those spawning populations are being absolutely hammered. And we know that their populations are under extreme pressure. So that was one of the reasons for the recommendations or the regulations that came out with the new MPAs, the coastal MPAs that were promulgated, is that no night fishing, boat-based night fishing is allowed. And that includes Protea, Alawal Shoal, Tugela, and Ismangaliso. So you're still allowed to fish from the shore at night, but for boat-based operations, there's no night fishing allowed. This was contested by the commercials last year or the year before, and they were won some relief whereby they were allowed to continue to fish at night because it's for the livelihood of the operations. But in terms of recreational fishing, there is no boat-based night fishing allowed in the MPAs, which I believe will go a long way to start to help protect those very important spawning aggregations. It's like killing the golden goose. You know, year, they aggregated on those areas, and catching them in those numbers just makes absolutely no sense. It's stupid.
Okay, thank you. Thank you very much, Bruce. And I think that I've just got 2 slides that I'd like to show. But before I do my final 2 slides, I'd like to say a very big thank you, Ryan and Bruce, for being prepared to do this. This is the first time that we've done it. So a very, very big thank you for being our first ORI presenters at a SAAMBR webinar. So thank you so much to the 2 of you. A very big thank you to Lynn. She handled all of the RSVP, sending everyone the links. So Lynn, a very big thank you to you. We don't have an automated system, so it's all done manually. So thank you very much, Lynn. And thank you to all of you for joining us for the last hour and a half of presentations. As somebody said in the chat, this is really what we need to do. So if we want to communicate more effectively about marine protected areas, we need things like Marine Protected Area Day, but we also need presentations like this that really share information about the protected areas. And then, as we've said, we need more research that looks at the social and economic effectiveness as well. So maybe we'll start a series. So I'm just going to very quickly share my screen because I do have to do this last little plug. For those of you who don't know, we have, can everyone see the screen?
Not yet.
Not yet. Hopefully it's coming. I wanted to just tell you a little bit about the Marine Fish Guide for Southern Africa. It's an app that Bruce has been working on for the last 2 years. It's got almost 250 marine fish species that are likely to be caught by fishermen around the coast of South Africa. It's incredibly informative. So if you haven't seen the app yet, please go along and have a look at the App Store. And then the last thing is, if you'd like to hear more about our research and you are one of those people who does social media, please have a look at one of our social media handles. So I think that there's nothing for me to say, but a huge big thank you so much to everybody. Have a lovely drink now, have a good supper, and thank you to our speakers. Thank you very much, everybody, and good night.